Hello everyone, my name is Emily Watson, and I decided to do my project on norovirus, but specifically norovirus in children under the age of 5 with gastroenteritis. Norovirus is a non-envelope positive stranded RNA virus. This type of virus belongs to the Calciviridae family. There are a total of 7 genogroups within that virus. However, the study that I have chosen mainly focuses on the genogroups G1 and G2 because they are the most common. This type of virus is very contagious and causes vomiting and diarrhea. There are 3 ways a person may become infected, which is having direct contact with an infected person, consuming contaminated food or water, and touching contaminated surfaces and putting unwashed hands in the mouth. Norovirus is a leading cause of human viral gastroenteritis globally, and responsible for more than half of gastroenteritis that occurs yearly. It is also the second largest contributor to severe childhood gastroenteritis in low-income countries. This is most likely due to the fact that underdeveloped and low-income countries do not have the best sanitation practices, and children are more likely to put their hands in their mouths. The study that I'm going to be talking about today looked at Cameroon, which is a country in Africa that sees about 14,400 deaths in children related to diarrheal disease. Here I have a screenshot of the article. The article is titled Molecular Epidemiology of Norovirus in Children Under 5 Years of Age with Acute Gastroenteritis in Yaoundé, Cameroon. Before I talk about some of the molecular methods that were used in this study, I first want to talk about how they went about choosing the sample population. They used the definition of gastroenteritis from the WHO and AFRO rotavirus surveillance protocol to determine which patients qualified. The exact definition was the occurrence of at least 3 looser than normal or watery stools in a 24-hour period and 2 or more episodes of vomiting unexplained by other reasons. A total of 2,831 stool samples were collected from January 2010 through December 2013, within 48 hours of admission from children below the age of 5 with acute diarrhea. Patients came from a total of different districts in Yaoundé, Cameroon, which I've listed below. Out of the 2,831 stool samples that were collected, 902 samples were randomly selected to test for the different genotypes of norovirus. The first molecular method that was used was reverse transcriptase PCR. This method was used in order to amplify the target RNA from the stool specimens. This slide outlines the exact procedure in which they used. I am not going to read the entire procedure, but if you would like, you can go ahead and pause and read it. The PCR products that they got from the transcriptase PCR were visualized on a 2% agarose gel, and amplicons of the expected size of G1 330 base pairs, and G2 341 base pairs were gel purified with QIA quick gel extraction kit and sequenced by Sanger sequencing. Other molecular methods were done, including phylogenetic analysis and genotyping. Phylogenetic analysis is when there's branching diagrams that are made to represent the relationships between different species, organisms, or characteristics of a certain organism, which could be genes, proteins, or organs. The diagram that is created is known as a phylogenetic tree. In this case, sequences were aligned and trimmed using assembler, a type of software that is used to store and analyze biological data, and the genotypes were determined using an unweighted pair A-group method with arithmetic mean phylogenetic tree and gene-bank norovirus reference sequences. 1,000 different replications were used to determine the significant differences between the strands. Genotyping was also performed in this study to determine the genotypes of the specimens that tested positive for norovirus. 2.6% of the specimens were G1 and 97.4% were G2. None of the samples were positive for both G1 and G2. G2.4 was the most common genotype, representing 60% of strands in 2010, 86.2% of strands in 2011, and 82% of strands in 2013. After all the molecular analysis was done on the samples, it was determined that out of the 902 selected stool samples, 8.4% of the specimens tested positive for norovirus. The majority of the positive samples, 89.5% of specimens, came from 4 locations, which I have also listed. The table on the right outlines all the districts included in this testing and the prevalence of norovirus in each district. The median age of children with norovirus-associated diarrhea was 7 months, with 97.4% being under 24 months of age. Statistical analysis was done to compare prevalence of norovirus and gastroenteritis in other parts of Africa to Cameroon. The p-score of greater than 0.05 shows that there was not a significant difference between Cameroon and other parts of Africa in prevalence of norovirus and gastroenteritis in children under the age of 5. To conclude, norovirus was detected in 8.4% of diarrheal stools from hospitals in children under the age of 5, with cases of acute gastroenteritis. These results are in the same range as results reported in a systematic review of norovirus in Africa. The data collected in the study and from the review previously done show that prevalence of norovirus in Africa is similar to the range reported globally. It was also concluded that G2 viruses were most frequent in cases in Africa and worldwide. And in figure 2, you can see the monthly distribution of norovirus genotypes detected from 2010 to 2013 in Yaoundé, Cameroon. And you can see that G2.4 was the most common, that is the green. There were several different study limitations that I thought was important to note. One of those being that they couldn't attribute norovirus to the cause of diarrhea because they did not test for other pathogens that could also cause a co-infection. A more robust study design would be needed to better represent the population in order to address the attributions and the norovirus incidence estimates in Yaoundé in children. Genetic sequencing was only performed in the capsid region for viruses. Detection of recombinant strains was not assessed, which can also be the source of norovirus epidemics. And only a portion of the samples collected were tested. All samples should have been tested to provide a clearer picture of norovirus incidence in Cameroon. This is the first study to investigate norovirus in Yaoundé, Cameroon. Larger-scale community and/or hospital-based epidemiological studies, as well as molecular surveillance, are needed to better characterize the incidence and molecular epidemiology of noroviruses in pediatric populations in Cameroon. More studies will provide more evidence to inform policymakers and healthcare professionals on the importance of noroviruses in diarrheal illness and death among children, which would also allow for the importance of improvement of prevention and control methods. These are my references. I just wanted to say thank you for listening to my presentation.